Aloha, thank you. Welcome back, and aloha people at home. Today, we have our second speaker of the morning, and I'm going to welcome Nalu to the floor. He is the director of the Kumuloa Marine Science Education Center. He has been an active researcher, educator, all around great person forever, but really intensely for the last 15 years. He is dedicated to both our seascapes and our landscapes, and dedicated to bringing forth that next generation of stewards for those areas and for our culture. And so Nalu, please come to the floor.
Thank you. Thanks for that introduction, Lisa. Whoa. I'm the generation that also skipped the ability to use the phone, so I have a printed out paper, and I didn't want to miss anything when I introduced Cienna because she's so important to us. So it's with great pride that I introduce Cienna-Lei Kuʻuileialohaiwiliʻia Daog to you as our program's first TCBES internship mentee, and now our close friend. Cienna-Lei proudly calls Keonipoko Puna her home. She's a 2017 graduate of our Kamehameha School's Hawaiʻi campus, as well as a 2021 graduate of Hilo and with a biology degree. Let's see, I know Cienna is a skilled lawaiʻa, a person deeply passionate about her ohana and her aina, a lover of dogs, and of good Filipino food. When I asked our coworkers to describe her, they used the words ha'a, olu olu, grounded, respectful, self-directed, funny, and easygoing. Cienna jumped right into our program and immediately led our Āhole work that she's about to share. But it's worth mentioning that she's done so many other things at our local i'a that were so important to how we function and raise the next generation. I could go on and on really about this resilient wahine mana here, I really could. But Cienna, I just wanted to say that our team could not be more proud of you. We could not be more honored to work alongside you, and we look forward to your career in Hawaii.
Wow. I might cry. Aloha mai kākou. My name is Cienna-Lei Daog, and today I will be sharing with you all my journey as a graduate student through my internship work on utilizing.
I'm trying. Sorry, technical difficulties.
Hi, Auntie Diane.
There we go. There we go.
My project is called Utilizing Science to Malama Aina, Evaluating the Fish Species Āhole as a Substitute for Introduced Species in Aquaculture. When Lisa approached us and asked us who we wanted to internship with, I knew I wanted to partner with an agency that reconnected me with my native Hawaiian culture, provided me a, sorry, my mom's in the crowd again. Provided me an Aina-based learning experience and gave me the opportunity to give back to Native Hawaiian community, which has led me to partner with Kumuloa Marine Science Education Center here under the big red dot. Kumuloa is directed under Kamehameha Schools, and it is located in the Iliaina or land section of Honolulu, which many of you know as Keaukaha, in the Moku or district of Hilo, and the Āhole land division of Waiakea. This is a pretty cool map that Michaelani provided for me. I think this is one of the first maps of Honolulu. Right, so here, I don't know if you can see the laser, but this is Kumuloa. Here, a broad range of community members, from students to researchers, come together to contribute to the restoration and rehabilitation of local i'a, or also known as Native Hawaiian fish ponds. Everyone that visits here carries out the kuleana, or responsibility, in perpetuating Native Hawaiian culture. here's just some of the students that come and visit us and get dirty in the sediment. The picture at the top right is a group from Volcano National Park. They were measuring sea level rise. The stewards of Kumuloa are Trish, Nalu, and Michaelani. I tried to convince them to come here in their outfits, but they refused, but they each play a vital role in maintaining the function and vitality of Kumuloa. And an interesting fact, they are also alumni of the TCBES program. Here are the local i'a located at Kumuloa. We have Āhole, Kapalaho, and Waiopio. Here you will see many native species such as the ama, the o'opu, Āhole, our nene birds, and alku'u. Local i'a is a native Hawaiian fish plant. There are 6 different types. However, the local umeiki, which is on the bottom in the middle, is known as a fish trap. The most common local i'a is local kuapa. Common characteristics of local i'a are kuapa, which is a rock wall, and makaha, which is a sluice gate. There are many functions of a local i'a. They provide habitat for many native species, which I mentioned before. During ancient Hawaiʻi, they provided food resources such as fish and kalo for native Hawaiian communities. There are many challenges that we face. Before Western contact, there were many local i'a throughout the Pai'aina, but as Hawaiʻi modernized, our lands developed. Here's a picture of just land development, downtown Hilo. And also an influx of introduced species came along with modernization, and invasive species are now a threat to our local i'a. So here we have California grass, which alters the sedimentation in the local i'a. Some invasive fish species you will see in local i'a are Mexican mollies, tilapia, and conda. For my internship project, I studied Āhole or Āhole, specifically the Kuhlia xenura species. The difference between Āhole and Āhole is their size. Āhole is their juvenile stage, which is a finger length, and Āhole is at their mature stage, which is a shaka. There are two species of Āhole found in Hawaii, Kuhlia xenura and Kuhlia sanvicensis. They are silver-bodied and they have really big eyes, as you can see in these pictures. The xenura species is endemic to Hawaii. There are many reasons why we chose to study Āhole. One of the main reasons connects to this saying, He mana ko kainoa, which means there is power or meaning within a name. Waiahole is one of the local i'a at Kumuloa, and Waiahole loosely translates to Āhole waters, which indicates that these areas thrived with Āhole. It also speaks on 2 important resources, which is Wai, freshwater, and Āhole. We cannot have one without the other. Waiahole also indicates that Āhole thrive in Waikai, which is a mixture of freshwater and seawater, also known as brackish water. So the salinity of freshwater, of course, is 0 parts per thousand, and the salinity level of seawater is 36 parts per thousand. Parts per thousand is the measurement of salt in water. and the salinity level at the local at Kumuloa is 4 parts per thousand. So contemporary aquaculture here in Hawaii utilize non-native and potentially invasive species, such as the tilapia pictured here on your right. They are becoming invasive to our native ecosystems. They're also a significant threat to our lokoia because they can thrive in multiple aquatic environments and they're quick reproducers. Here is just an aquaculture farm with tilapia, just showing how robust they are. I can't even count how many are in these pots. So knowing that Āhole thrive in lokoia, which is a low salinity environment, and knowing that tilapia are in a freshwater environment that is essentially going into our natural ecosystems, leads us to the question if we can implement Āhole into aquaculture systems. So seeing if Āhole can survive and thrive in aquaculture systems. So during my time at Kumuloa, I had 3 projects. So the first trial was, can Āhole live in fresh water? The second trial was, what salinities do Āhole thrive in? And the third were, can Āhole be candidates for aquaculture and aquaponic systems? So here is trial 1, can Āhole live in fresh water? The purpose of this study was to measure the growth rate of Āhole in a controlled environment and a treatment environment. So the control groups remained at 4 parts per thousand, which is the salinity of the lokoia. And for the treatment groups, I lowered the salinity all the way down to freshwater and just measured their growth rate. We did this experiment twice. The only difference was the water in the treatment tanks. In the first trial, we lowered the salinity with rainwater, and in the second trial, we lowered the salinity with rainwater with an added buffer solution to stabilize the alkalinity. Here is the design. As you can see, we used 10-gallon aquarium tanks. In each tank, there are 3 Āhole. I lowered the salinity by 1 part per thousand each week, and I also measured the growth rate as I dropped the salinity. This project took place over 4 months. Here's the results of trial 1. For the growth rates, the bottom is the first trial with just rainwater, and at the top is our trial 2 with rainwater and the buffer solution. And then we have the body mass percent change from start. So looking at these red arrows, that indicates that there was mortality in both the fresh water treatment tanks. So at day 20 and at day 65, all the IA in the treatment tanks died, which concluded the project. This graph also shows that the growth rates in both control tanks are higher than those in the treatment tanks. For the body mass percent change from start, also known as doubling time, which is when the fish doubles in size, and it doubles in size when it surpasses the 100% change. So for juvenile Āhole, it takes approximately 42 days and 4 parts per thousand to double in size. So now that we know Āhole need some sort of salinity environment to thrive, the next step was to study what salinities do they best thrive at. So for this trial, we had 8th graders at Kamehameha schools replicate trial 1. However, they measured the growth rate at 1, 2.5, 4, 7, 10, and 17. They did the same exact thing. They just measured the growth rate over time. So here are the 8th graders. They had 10-gallon tanks. They also had 3 ʻaholehole, but the ʻaholehole that they used were a little bit bigger than the ones in the first trial. And as you can see, they are collecting the growth rate, making sure they put the right fish back in the right salinity tank. Yeah, it's pretty cool to see this project spread throughout, not just me. So yeah. So here are the results from trial 2. Again, we have the growth rate and the body mass percent change, or doubling time. From the graph of growth rates, we can see that all the ʻaholehole grew at their tested salinities. The larger ʻaholehole did display aggression. So in the beginning, they started with 3 i'a fish. Sorry, I should have mentioned i'a is fish. They started with 3, and then they ended with 1. So there were 1 fish that remained in each of the tanks because they killed each other. Yeah. So for the body mass percent change, our quickest doubling time was at 4 parts per thousand at roughly 99 days, and the slowest doubling time was at 16 parts per thousand, which is about 252 days. So knowing that the quickest doubling time is in 4 parts per thousand, we enter trial 3, which is, can we integrate ʻaHole into aquaculture and aquaponic system that is at a salinity of 4 parts per thousand? This was by far my favorite project because we got to build an aquaculture system from the ground up. Here we used a 40-gallon tank instead of a small 10-gallon aquarium tank. In this picture, I created water pumps out of PVC pipes. And then here you can see Luke, Nalu, and Waikalani helping me build. Well, I forget what we were building, but we were building something. In this second picture, or the video, I created the water filter with a water bottle. I added filtration pads and biofilter media, which grew healthy bacteria. So as the water flowed through the pump down into the filter, we picked up all the gunk, and then the water became nutrients as it passed through the biomedia. Once the system was complete, we added water from the local i'a because it is 4 parts per thousand, and we also sourced our ʻaHole from the local i'a as well. We then domesticated them using a commercial pellet feed called Aquamax, which was provided by Matt. So here are the results from our 3rd trial. As you can see in the growth rates, all ʻaHole were growing, which means that our conversion to commercial feed was successful because they were growing. We had 8 ʻaHole in our 40-gallon tank, and we found no aggression, which is a good sign because we do not want them to kill themselves. For body mass percent change, doubling time, we found the range between 374 days and 529 days. Keep in mind, these are ʻaHole, so they're already at their mature stage. They're not ʻaHole, so that is why the range is quite large. Here are ongoing projects. We have been tagging ʻaHole from the local i'a, basically a catch and release system, to measure their growth rate in their natural habitat. And also, we are in the middle of transitioning the aquaculture system into an aquaponic system. So taking the ʻaʻole from the aquaculture system and putting them into this aquaponic system, and that's playing in the video, and then implementing salt tolerating plants. Just a brief summary. Trial 1, can ʻaʻole thrive in fresh water? Our study suggested no, but they do thrive in 1 part per thousand. For trial 2, what salinity do ʻaʻole best thrive in? 4 parts per thousand, which goes all the way back to he mana kōkua inoa by ʻaʻole, just proving that our kupuna were right. And for trial 3, can ʻaʻole be integrated into aquaculture aquaponics? The answer is yes. However, we have to take in consideration the tank size and the growth rate for the stage of the ʻaʻole or ʻaʻole. This project would have not been successful without all these people. Thank you to Lisa, Nalu, Trisha, Michaelani, Dr. Armando, Sydney, Dr. E and his 8th graders, my family, and my cohort. Thank you for the love and support over these crazy 2 years. Mahalo.
Okay, so we have time for questions, and so if you have a question in the chat, you can just write it in the chat, or you can speak it out loud. So are there any questions? Okay, Alejandro.
Yeah, so in that first trial where you had that freshwater treatment, you said that those fish died during those trials. Was that like a gradual process that you noticed a decline in their health, or was it very sudden?
Yeah, no, it was gradual. They died 1 by 1. So there was no aggression. We didn't see any aggression in the tanks. It just shows that they do need some salinity to survive.
Do you know approximately how many fish traditionally have been in these or are currently in the local i'a? And because aggression was a problem, right? So I'm wondering if in nature they're usually a bit more spread out, or do you know about how many fish would be?
I do not know.
Okay, that's okay.
I can go count for you though.
Yeah.
I was curious, why do you think you saw aggression in the smaller, like 10-gallon tanks, but not in the 40-gallon tanks?
The first thing that came to my mind was that they are taken out of their natural habitats and put into such confined spaces. So it's like a fight or flight situation. So in this case, they all fought.
Yeah. That makes sense.
Yes. No.
Can you repeat the question for the audience at home?
Oh, can this, do we do this
I'm
trial?
doing the same thing with Packer, with and they have been getting to 0. So it was a GH problem. And you got another like 1.9. The general hardness was the general hardness in the brackish is 22. Coast water out of city water is like 3, so we get count. But I'm wondering if xenura. I just started to xenura. So I'm wondering if we also have this problem, not just with the salt content.
Yeah, we believe that alkalinity was why they died in the first trial. We tried with Sydney's buffer. Because this,
I wouldn't stop.
Yeah.
I think you get it out there.
Yeah,
I think we're right on the verge of getting it out. Then you have to be ready more plants. That won't allow you.
Right. Yeah, that is the goal. Hopefully we can get them to freshwater. That would be amazing. Yes.
What are the advantages of having it like freshwater opposed to just using like a little bit of like 4 parts per million?
So many plants do not tolerate salt water. So when you get Āhole into fresh water, that opens up a more variety of plants that we can propagate in the aquaponic system. So for example, because we found that they thrive at 4 parts per thousand, we are planning to implement sea asparagus, like olukai. Are you familiar with that? No. That's a salt tolerating plant.
Alkalinity affect the fish?
I'm not an expert in water.
Do you know like what part of the alkalinity?
The other way is just above there, it's the heat. Say, the G, that's the KH, the GH is the general, that's magnesium, and calcium, that sort of stuff.
So it's more like those.
That's what makes water hard or soft. That's what, soft water on the way, or no, we have soft water here out of the hose, because we don't have, soft tanks, we have a lot of calcium carbonate. It's calcium and magnesium. So I found the sense of that's what they were missing.
I have a question.
Yeah.
Do you have any future plans to go back to commitment schools with other projects?
Oh, I would love to. Yeah. My mom is here. She is a kindergarten teacher at Kumuloa schools, and they plan to implement the aquaponic system into her classroom.
Oh, very fun.
Yeah, pretty cool.
All right, well, we're at time. So I just want to thank you, Cienna, for your. All right, and so we're going to take a